Hello.
Hi.
How is everybody today? Happy?
All right.
Good to hear. Oh, Julia's here. Hello, Julia. So good to hear from you. Michelle Bello Veramendi. The Veremenster. I like to call her. Here we come. We're recording this, by the way. Oh, you can't see me. My security mechanism was engaged there. I was put black tape over my camera because there are certain viruses that'll turn your camera on remotely. Gotta be careful. Okay. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Iorio's here. Life is good. Iorio, 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 Iorio. Okay, so I recorded our last session, our Monday session, and I put that up on Moodle, so you can look at that again. It's pretty bad, frankly, because there's a whole lot of downtime. The first 5 minutes, I'm just waiting for people to show. So you got to skip through a bit, but it is up there, and I will be doing that again with all our Zooms from here on out. So, couple of announcements. Hello, iPhone. iPhone's here. What were my announcements? Oh, one is that today's lecture is going to be a little short. There isn't all that much information here. We're talking about neural crest cells. We'll talk about where they're from and where they move and what they become. Not too much detail, though. We're not getting into too much detail. I'm just kind of mainly focusing on signals and what the signaling pathways are. The other thing is, I notice after today, we've only got 4 more lectures. We've got next week and the week after, and then it's finals week. So if we bring up the, let me share. Share screen. Share this. We'll share this first. We look at our syllabus. We still have to do chapter 7, 8, 9, 11, and 14. So we're going to have to start moving a little bit faster so we can get through all of that. If you guys don't mind. Everybody okay with that? These are the jokes, people. This is where you laugh. Slish. Okay, so clearly we're not going to go through all that. We should be able to finish chapter 5 on Tuesday. So I'm going to look through the syllabus and the book and see what's most important, and we'll cover some of that for the last 3 lectures. And then we'll have our 4th exam during finals week, right? No cumulative final, just a 4th exam.
Dr. Slish, quick question.
Go.
Well, it's actually a statement, I guess. I clicked on the last class's YouTube Zoom thing upload,
Yeah.
and it said it was like private, so I couldn't view it. I don't know if you have to change the privacy settings to allow us to, but I couldn't access it.
Oh, okay. I thought if I gave you the link, you could get it, but you couldn't search for it. So I'll change that to make that accessible. Sorry, thanks for telling me.
Thank you.
All right, let me pull up the chat just so that I can see if anyone has questions. So cumulative final, only for you, Mr. iPhone. Everyone else has no cumulative final. Just gonna be a 4th exam. How do I move this? This son bitch. I'm trying to move the chat out of the way so I can see my my PowerPoint while talking about it. Okay, are we ready to rock here? So today we're talking about neural crest cells and the origin of neural crest. We talked about this before. I looked through the old PowerPoints. I couldn't find where we had seen it before, but we have talked about them before. They're migratory cells. They go to a lot of different places in the body, and they're really important for setting up different things. So that's what we'll talk about today. So this slide is slide 31. So in the notes, let me pull up the notes real quick. The very first thing in the notes is a link to a YouTube video, and it is really good. It's this woman from Barcelona, and she explains things really well, I think. And her figures are a lot better than mine, or the ones from the book. So I'm going to go through this real quickly with the ones from the book, and then we'll look at neurulation again, or the neural tube formation with her figures, because they're a lot better. Okay, so if we look at slide 31, here is your neural plate. And remember, we're going to be talking chick, mouse, same thing, I guess. As the node is coming back, you're laying down your Hox genes, and cells are going in through the node and becoming the neural plate and the nodal cord underneath. So you're making mesenchyme and neural cells as that node is regressing from the anterior to the posterior side. So we're starting after the regression has happened. And so you've got neural plate. On top of the neural plate, there's epidermis. And then you've got lateral mesoderm. Well, actually, this is all that ectoderm. So now it's neural plate, and then here's the epidermis on the outside, the gray part. Okay, so once that neural plate forms, it starts to invaginate here, and you end up with the neural groove. The neural, oh, hang on. Ooh, look at that, that moves. So here at the margin between the non-neural ectoderm and the neural ectoderm is where is the origin of your neural crest cells. So as this invaginates, that neural crest cell ends up on the outside of these neural folds, and then as it folds up, you get your neural tube, and then at the dorsal side of the neural tube is your neural crest. Now this figure, I don't really like comparing it to her figure because hers is a lot nicer. This is showing neural crest cells on top here, and they're migrating out, which is true, but also this whole dorsal side of the neural tube is considered neural crest as well. So keep that in mind. So we'll look at the figures then from that animation, which you should definitely watch. Here we are again at the neural plate stage, and you got your nodal cord, neuroectoderm, non-neural ectoderm, and then you get induction of the neural crest cells at this border between the non-neural and the neural ectoderm. You get the folding, neural groove, the neural ectoderm cells come together, and neural tube seals up, and now these non-neural ectoderm, which become epithelium, fuse up above it, and now you've got your dorsal part of the neuroectoderm. Neuroectoderm fuses here, and that's your neural crest. And at this point, then, those neural crest cells start to divide and multiply, so you get more and more of them, and then they start migrating away. And they're going to migrate not anterior or posterior, but pretty much just laterally and ventrally. So they're starting all dorsal, so they're naturally going to end up going ventral, and also laterally out the other sides. Not so much anterior or posterior. And we'll talk about that in a little while today. Okay, so that's kind of the motions, which we kind of talked about before, but that's a bit of a review. Now we'll talk about the induction events that happen here that caused this to happen. So if we back up to the early stage here, if you remember, these cells are coming through that neural groove and differentiating and going out, becoming neuro versus mesenteric. That movement and the differentiation is all FGF and Wnt, right? We've talked about this over and over again in this chapter. FGF and Wnt is causing all that to happen. So once they come through and go out, they're exposed to a high concentration of FGF and Wnt, which is being produced in that area. Now, the other important signaling molecule we always talk about is BMP4. So BMP4 is coming from laterally. It's coming from the lateral mesoderm, I think, or some cells that are laterally anyway. So it's coming from the side in. So whenever you have, a point source like that, you end up with a gradient of BMP activity. So BMP is going to stop the spread of neuroectoderm. So it's limiting how big that neural plate's going to be. So you get a sharp demarcation here. But, and then when you do have a lot of BMP, that ectoderm stays neural over here. But what you end up doing because of that gradient is you've got an area with no BMP4, which is neural plate, and then you've got an area with high BMP4, which is non-neural ectoderm. But in between the two, you've still got BMP4, but it's very low. So you've got a low concentration of BMP4. And because you have that low concentration of BMP4, you change what genes are being activated. So what we get activated there at that border is SOX9 and SOX10. And those transcription factors convert that non-neural ectoderm into neural crest cells. So they're neural. SOX, remember from our Xenobus lecture, is a neural marker. So SOX9 and 10 are making them neural, but they're not as neural as the neural plate are. They're a little bit different. Now, as that groove starts to form, and then it seals up, and now totally seals, and now you've got your neural crest cells up here on the dorsal side of the neural tube, and now they're right next to this non-neural ectoderm. So here's a high source, excuse me, high source of BMP4. So now these neuroectoderm cells are going to be exposed to FGF, Wnt, and a higher concentration of BMP4. So we're changing again the signaling pathways that are being turned on, which is turning on different transcription factors, which is changing the cell type again. So here, once you get that higher concentration of BMP4, you start turning on two other transcription factors, slug and twist. So slug and twist then convert these neural crest cells into mesenchyme cells. Remember, mesenchyme is a general term for loosely organized migratory cells. So when those cells were coming backing up again during the regression of the node, those cells going in and becoming neural plate and nodal cord and somites, those were mesenchyme cells that then converted back into less migratory cells. Here, these are going from non-migratory cells into migratory cells. So that's what this EMT stands for here, epithelial to mesenchymal transition. So these are transitioning into a mesenchymal cell type. And when they do, they become, they let go of their cadherins, so they're not holding other cells anymore, and they start crawling away. And they're crawling laterally. So just to kind of sum up everything I said in 100 words, in 2 words, or bad estimate, but something like that. At this border between the neuroectoderm and the non-neuroectoderm, you've got FGF and Wnt signaling there closer to the neuroectoderm. Then you've got BMP4 coming from the sides. So right at that margin, you've got high FGF and Wnt and low BMP4, and that converts those cells into neural crest cells. And as that thing closes up, you get a higher concentration of BMP4, that turns on slug and twist, and then those cells become mesenchymal and start crawling away. Does that make sense? Everybody good with that?
Yes.
Yes.
Good. Okay. It's important to give me feedback because, you know, when we're not in class, I can't, when we're in class, I can see you. So I can't see you when we're not in class. Okay. So what do these things become? Thank you. Thank you for the feedback, everybody. Okay. So what do these cells become? So if we're on slide 37 now, you've got these uncommitted neural crest cells. They go away. And as we'll talk about in a minute, they've got Hox genes in them. So they are a little bit, faded already by those Hox genes. But when they get off into their position and they terminally differentiate, they become damn near everything. Okay, melanocytes, I've said that before. They give you the color of your skin. They become glial cells, which are helper cells for neurons. They can be the non-myelinating glia or the like Schwann cells that put myelin around the axons. They can become sensory neurons. They become neurons of the sympathetic nervous system. You've got your adrenergic chromaffin cells, which are adrenal medulla, right? The middle of the adrenal gland that produces epinephrine. And they also become the sympathetic neurons, which are adrenergic and cholinergic sympathetic neurons. Okay, here's a question, pharmacology question, for 5,000 bonus points. What are the only sympathetic neurons that release acetylcholine instead of norepinephrine? 5,000 bonus points. You can ace this class right now just by knowing the answer to that question. Anyone know?
Question?
What?
Can you repeat the question?
Oh, sure. Which are the only sympathetic neurons that release acetylcholine instead of norepinephrine? Nobody knows. I know. I have a degree in cardiovascular pharmacology. I do. Sweat glands. Sweat glands are sympathetic neurons, and they release acetylcholine, which is rather paradoxical. All right, they can also become parasympathetic neurons. So pretty much your whole autonomic system here is being made by neural crest cells. And parasympathetic, as you all know, are cholinergic. They release acetylcholine. Enteric, the neurons leading to the digestive system, which release serotonin, and also peptidergic neurons. And they become cats. Cats, get out of here. And cartilage. This is a lot of what they become, especially in the face. We'll talk about that shortly. They become cartilaginous, and then they can become other structures in the body after that. Okay. And here's another. I want to make sure that you memorize everything on this slide, just to give you something to do. I mean, we're all bored. No, I'm not going to expect you to memorize any of this, but kind of the point of this slide, slide 38, is that, you know, you can see segments in this slide on the left-hand side there. Make it bigger. You can see the segmentation, and you know where that's coming from. That's the somites, which are segmented and specified using the Hox genes. And so what those neural crest cells become is determined both by the Hox genes that they're expressing, because they came out, you know, along with all the other cells during that regression, but also where they end up, because there'll be cell-cell interactions that'll help them become what they need to become, but they are somewhat pre-patterned by having those Hox genes expressed in them. They maintain their Hox gene expression even as they migrate and go into position. What we'll talk about now is up here, the first few, this is even before the somites, the first few neural crest cells will come out and make a lot of the facial structures. So they're going to differentiate from that kind of neural crest, which is somewhat neural, into a more mesenchymal cell type and become chondrocytes and bone cells and all this stuff to make the facial structures. Okay, so point being here, they've got their Hox genes expressing, they're going laterally. So really within that segmented structure, they become what they should be in those positions. And here's an example of that. So what we're showing here, this big bump here are the gill arches, right? If you were going to be a fish, they would be these cartilaginous things that hold the gills in place. So when the water flows through, you can collect oxygen. You, me, we're not fish. So in evolution over time, these gill arches became other things. In mammalian, I think in bird too, they call them pharyngeal arches. They're going to become pretty much from here down. So you've got your lower jaw, your ear bones, hyoid bone, and other structures in here. So if we go back to slide 39, what they're showing here is how the Hox genes are going to specify to a certain extent what the neural crest cells will be when they get into place. Now here's a new vocabulary word, rhombomere. So rhombomeres are kind of segmentations in the nodal, or not the nodal cord, in the neural tube. So honestly, I'm not sure if they actually form nodes or structures. They don't separate, right? Because the neural tube is one constant thing, but I think they pucker a little bit so you can see differences. I'm kind of unclear on that. But one thing I can tell you is that these rhombomeres represent different areas of Hox gene expression. So, and they're numbered R1 through here is 8. So these rhombomeres are before the spinal cord starts. And where the spinal cord starts, I think that's where your somites start. I think actually the somites start in the hindbrain area, which would be over here, R7, R8. But anyway, instead of telling you what I don't know, let me tell you what I do know. So rhombomere 2, 3, 4, 5. These neural crest cells are going to come off the spinal cord or the neural tube here and go out into this face area and then differentiate into those face tissues. And the next two slides are an experiment that shows that they do have Hox gene expression and they are somewhat pre-patterned. So here's what normally happens. Your R2 neural crest cells coming from the R2 will come out into this first brachial arch, or the first pharyngeal arch, and that will then become jaw. Over here, the R4 rhomboid, sorry, my internet was horrible last few days. I keep getting kicked off too. Okay, so the neural crest cells from R4 will come out and become the second brachial arch, which then becomes things in the throat here. So one way to show that well, we know these cells are pretty multipotent. They can become a lot of different things based on what they're expressed on, but they do have some pre-patterning, because if you take R2 cells out and you remove the R4 cells and you put the R2s in the R4 space. Okay, so here we've taken R2, we've gotten rid of R4 and put R2 there, and they come out here, and the neural crest cells come out, and you end up with, you do this in chick, end up with a chick that's got two jaws. So instead of a throat, it gets a jaw, another jaw. So they are somewhat pre-patterned, although again, they are pretty multipotent. So let me let you digest that for a minute while I let the dog out. Oh, yeah. It's funny, whenever I let him out, he goes out and he looks back like, "You're leaving me?" He expects me to go out too. Cold out there. I don't have a fur coat. All right, that's about all I have for today. Are there any questions about this? Any?
All good.
What's that?
All is good.
Everybody happy? Everything clear for me. Good. Christina's on it. Everything's clear. Yeah, I don't think this one was that complicated, not like some of the earlier ones. So I may close this down now if everybody's happy.
I'm good.
Everybody's good? Okay, pretty simple. Good. Yeah, this one wasn't a very complicated lecture. So the next lecture we will do, what is the next lecture? Oh, left-right patterning. So if you remember, lefty was really big back in the first half of the chapter, and you're going to see another appearance of lefty here, and we're going to get some left-right patterning. Yay, yay, left-right. No questions? Okay, I'm going to call it a day then. Thank you so much for coming.
Thank you, Dr. Slish.
Thank you.
Thank you.
Take care.
So I'll post this probably today, okay? And then you can look at it again later.
Okay.
I'm good.
Okay, later, dudes.